I'm Tanya. I'm the Client Success Lead at Adres. This is our very first webinar, and we are happy that you are here with us today. So I'll say a few words about Adres, and then we will start our session. Adres is an international consultancy company. We are working with startups in the biopharma field. We have clients all over the world, in the US, Europe, and Asia, and of course, Israel. Adres provides consultancy and outsourced services in regulatory affairs, QA, and CMC. And we see a great value in sharing our consultants' knowledge and experience with the biopharma community. So today I have with me here in the office Or Degany, our CMC director, and he will talk about process validation. The session will take an hour, and in the end, we will save 10 minutes for questions. Because this is a broadcast, a live broadcast, we can't be able to answer questions during the session, so we'll approach it in the end. So feel free to write your questions in the chat. And one last thing before Or take the stage, I will share a link of Google Forms in the chat, so you can fill in your name, title, and contact info, so we can get in touch after the webinar. Thank you very much. Enjoy, and Or will take it from here.
So hello to everyone. My name is Or Degany. I'm CMC Director at Adres. Today, sitting with me in the office, you already know Tanya, which is our client's lead success. And also Dr. Tamar Oved, our QA Director, is also joining us today in the room. We're very happy that you are joining us today in our first webinar. And as I said, as Tanya said, we will go through a presentation and hopefully have a few minutes, 5 to 10 minutes for questions. We're very excited for you joining this webinar, and we really hope that you will find it useful. So thank you very much. So today's webinar is about approaching rather than bumping into process validation. In general, the discussion will be about awareness, about being prepared, about planning things in advance, because process validation is a very critical step or milestone in a company's clinical development program, and you really want to reach it while you are ready for it, and you do not want to approach it while you are surprised. You need to catch up on a lot of things that you did not do historically, and then you want to start process validation in June, and you find yourself starting it a year later. And these are issues that we encounter, and that is the reason why we thought it would be useful to providing this training. So, when is the right time to start preparing for process validation? So if we look on this classical scheme that shows the classical phase 1, phase 2, phase 3 BLA filing program, we can see on the bar below the 3 stages of process validation. The first stage is the process design. The second stage is the process qualification or process validation. And the third step is the continuous process verification. We will mainly talk today about preparations for stage 2, which is the process qualification or the process validation. And getting prepared for process validation is actually something that needs to start during stage 1 of the process design. that runs in parallel to phase 1 and phase 2 of your clinical trials. So as you proceed in phase 1 and from phase 1 to phase 2, and during phase 2, you will have to do many activities, some in tandem and some in parallel. So you are able to come to a situation where finally, when you finish phase 2, you are relatively ready. You are relatively ready for phase 3, and in most of the sequences, the process qualification or process validation is being run in parallel to phase 3. So what we are going to talk today is how can we manage and plan ahead of time, so we're going to have a relatively smooth transition from finishing the stage 1 of process design and going to the stage 2 of process qualification. So from a conceptual point of view, we want to have a mindset. And this mindset is talking about being aware ahead of time, planning issues, prioritizing issues, and acting in a phase-appropriate manner. If we're going to look on this scheme of this roadmap on the left side, we are here today in the red dot in the process design, and we want to approach process validation and not to be in a situation that each time that we make the progress from process design to process validation, we meet a lot of bumpers. And these bumpers are there because we weren't aware of them a priori. We didn't plan when we will meet them and how we will meet them, and we didn't prioritize. And prioritize is meaning what is critical, what is less critical, which issues have a long lead time and we want to tackle them first, and which issues are short lead times and we can delay dealing with them to a later phase. And in addition, very critical is to always define what is your critical path. What is the activity which, if you will have delays in that activity, it will delay your introduction or your start of process validation. So on the end of the day, this is the message that we will try to communicate today: awareness, planning, prioritization, phase-appropriate development, and define of critical path. So another conceptual slide here is regarding the fact that process validation is really your money time of your process. There are a lot of activities that have to be ready and set in place before you start process validation. So this is like a funnel. It's like a critical time point in your development where many, many issues have to be already ready before you commence process validation. And some of them are written here, like you have to finish your process development, your quality by design, you have to have specifications defined and justified, you have to have your release tests, you have to have some accumulated data already on stability, analytical methods in place, validated, a lot of activities. So this is your money time, and it's better that you're ready for this money time event and you don't want to encounter a situation where you come into a process validation and you give your board, your management a commitment, and when the time comes to commence with process validation, there are a lot of things that are missing. So one of the most important, I'm sorry about the gibberish in the product here, but one of the most important issues in doing process validation in biologics is the phrase that the product is the process and the process is the product. This understanding is very, very critical to understand. The way that regulators look on biopharmaceutical products, because these are very complex molecules, it is not enough just to have all your release tests conforming, because there are many things that we cannot check, or it's very difficult to check everything for such a complex molecule to see that everything is in the right place. And because of the sensitivity of a biomolecule to different changes in the process, it is very critical that each time that you do your manufacturing process, it will be done in the same way, because differences in the production process can result in effects on the attributes that you have for your biological molecule. And that is the reason why the term says why we say the process is the product and the product is the process. So it's not only your release tests and final product that count here, it is your process, understanding your process, controlling your process, and working in a repeatable manner. So, at first, we want to talk about a few things in the stage one, in the process design. As we said, process design is happening in parallel to your phase one and the phase two studies. And according to our experience, there are a few issues that we would want to highlight that we sometimes in the past saw that startup companies tend to be less aware of or should provide more attention to. And we want to provide you a few examples of these issues so you could be really aware of them. So first, during the process design phase, characterization tests of your drug substance are very important. These are additional in-depth tests for your drug substance, so you really know your drug substance in a more intimate manner. These tests are very, very important when you scale up and you tech transfer your drug substance manufacturing process. And some companies tend to be less aware of this, but it is an important issue. You should have a reference standard that is qualified, and this is also very important, as reference standard are a key tool in your analytical methods qualification and validation. You have to identify and characterize your impurities. You have two types of impurities. These could be process-related impurities, like endotoxins, like residual solvents, like bacterial bioburden, and they could be product-related, meaning derivatives of your product. And the guidelines are relatively very clear regarding what are the levels that you need to report and identify when you have product-related impurities for small molecules, not biologics. And for biologics, which are large molecules, these are limits that the manufacturer needs to establish for himself. So very important issue, know your impurities. Remember, impurities are the place where safety and CMC go hand in hand, and it's very important that you understand your impurity profile. Forced degradation studies. Okay, why do we need that? You set out a stability protocol. You have to make sure that your methods are able to see the degradation or changes in your molecule, because if you will not provide evidence of that, in theory, your methods could be blind to changes of your product during stability. So that also is a very important issue. Make sure that you have enough IPC tests in each stage and do not make that too lean. So in each process intermediate step, you need to have 2 or 3 or 4 IPC tests with a rationale why you use these tests. And please remember, IPC tests are your way of ensuring that on the end of the day, you will click mark all the correct stations on the way, and you will reach a drug substance with the correct quality attributes. Another very, very painful issue is cost of goods. Remember that you are selling a commercial product on the end of the day, and its profit margin can bear a certain amount of costs on it. Meaning, for example, if you use a hugely costly resin, you have to understand that this is something that you could afford. And why am I telling you this issue? Because startups run very fast. They want to show that they're able to perform, to have investors help them and raise money. And on the end of the day, they are stuck with these equipments or resins or technologies that are very, very expensive. They go into validation, and then it's a little bit too late to optimize the cost of goods issue. And this I'm talking out of even my personal experience. So please take that into account. Always look on the cost of goods related to your manufacturing. Build hold times into your manufacturing process where appropriate. Please remember, a manufacturing process is not always perfect. Sometimes the lyophilizer could break down. Sometimes the drug product filling machine could have an issue of maintenance that needs 7 or 8 hours. So risk assess the places where you could have a stop in your process and build into those certain places or intermediates, build hold times inside and prove that if you hold your process 12 hours before drug product filling, it's okay. And maybe you need to prove that if you're waiting for an analytical result 12 hours, you need to build a hold time of 12 hours to show that your product is still stable while you are waiting for that analytical test. Follow change control practices. Very, very important to be in a control situation where you change issues. You don't want to be in a situation where you change things in your manufacturing process and you lose control on the process. And an additional issue that we want to focus on for biologics is make sure that your master cell banks and working cell banks are tested, are produced in a GMP manner, and you have all the documentation for this GMP production, and you commence stability studies for your master cell bank and working cell banks. Know your host cell line history. What is the source of your host? How the transfection was done? How clone selection was done, and document everything. On the end of the day, documentation is very, very critical. Okay, so it's very critical to document everything from the beginning, because if you think you can do it retrospectively, you're wrong. And you can review Model 3, which is a regulatory document, to see what are the things that would be expected from you from the regulatory authorities, and do that today. Don't do that in 2, 3, 4 years from today. So you know what is expected, and this is what we meant when we talked about awareness. So now we're going to go into some more specific issues. Process development report. What's a process development report? Very simple. Document what was done, when it was done, why it was done, and what were your conclusions. You can work with laboratory notebooks, that's great, but please summarize your key studies and findings on, computer files, on Word documents, and they will be very, very useful, because on the end of the day, when you will want to compose your final process development report, you will collect or fetch the key 10 or 15 files into one document, and very quickly you will have your process development report, which will provide the rationale for how you develop your process. Next issue is defining process parameter ranges. So why are we talking about this issue? So as you move along in developing your process, you will have ranges of parameters, the pH in your fermentation, the transmembrane pressure during TFF or hollow fiber, and these issues develop and evolve through the stages of development. Many companies tend to work with one single discrete values. They say we work at pH 7. We work at a flow rate of 350 centimeters per hour. We work at a TMP pressure of 0.5 bars, but that is not life. In life, you work in a range, and this is also this range is taking into account the operational and instrumental variation, the precision and calibration limits of your instrumentation, and it will provide you flexibility and act as a key component of your process control strategy. So please remember your parameters, your process parameters have to be in ranges, okay? And this is very important. Another terminology issue that we can see here in this scheme below is that we are talking about, let's get acquainted with a few definitions. So PAR, this is your proven acceptable range. It is the range while you are in it, your process will conform and you will have no effects on any quality attributes of your product. So let's say it could be, in this case, your PAR, let's say your target pH is 7. Your PAR can be between 6.5 and 7.5, but in the master batch record, you will ask your operators to work between pH of 6.8 and 7.3. So this is your NOR, your normal operating ranges. This is what your operators in production will see. PAR is the proven acceptable ranges that were proved to be acceptable by R&D. And out of the PAR, you will probably be in the range of failure. So please be aware of these basic definitions of PAR and NOR. The same goes with your IPCs, your in-process controls. As in previous cycle, your in-process control ranges evolve throughout the development. And also here, you need to work with ranges. Again, taking into account operational and instrumentational variations. We talked about precision and calibration limits. Again, so the important word, work with ranges, because that is life. Next thing is establishing your release specification and justified ranges. On the end of the day, you're going to make a product. This product is going to need specifications. So you would need to know what are your specifications for releasing the product and what are the ranges of those specifications. So you'll have like a grocery list of all of your tests that are related to the quality of your drug substance and drug product, okay? Mainly the safety, efficacy, and purity. You will always find these three words coming together in regulatory guidelines: safety, efficacy, and purity. And these are the issues that should be answered by your specifications. And you would need, of course, a rationale. And I'm repeating myself, but I will repeat myself again. Why you decided to have these list of tests and why did you decide to have these ranges? Some of the tests and some of the ranges are predetermined already by regulatory guidelines, and some of them are tests and ranges that you, as manufacturers, will need to set them up. And also, your specification will be a critical and key pillar of your process control strategy. Next issue that we want to talk about is the risk management. So what is a risk assessment or what is a risk management? One of the most basic tools of clinical development is using risk management methodologies. Many times in your clinical development, you will find yourself in places where you maybe could find yourself not spending a lot of money on experimenting things, on making data, but you have enough rationale, already have data, a common knowledge, scientific knowledge to solve things in a risk assessment by writing a risk assessment. And the whole way of thinking should be a risk management approach. You all the time think about what are the risks that are inherent in the things you're doing, and you want to manage them. And this issue of risk assessment strategy and risk management strategy is also correct when we're talking about process. And when risk management meets process, that's a definition that is called, we call it FMEA, or failure mode effect analysis, which actually is looking on your manufacturing process and asking what could go wrong. So you take each process unit, you look deeply into that process unit. It could be a fermentation, it could be a hollow fiber process unit, it could be a column chromatography, it could be a centrifugation, and then you ask yourself what could go wrong. You raise risks and you rate them. And those risks that will be considered high risks, and a high risk is also a risk that could affect your safety or efficacy or purity, that risk will be a potential critical process parameter. And please remember that because it will interconnect to the next slides. The FMEA document is expected to be a living document that is supposed to be updated frequently. And it's not, I would say, it's not a dead document, something that is a live document, and you are expected to update it from time to time. If you need more answers on how it looks and how to perform it, you are welcome to approach us. This is something that we do very frequently, and this is something that should be done frequently at these stages. The next issue that we want to talk about is that you need to start thinking towards the end of process design and before you go into process validation about having an established and representative scale-down model. What is a scale-down model? Well, you have a commercial scale process running in-house or at the CMO, and now you want to mimic it in a small scale, and not you only you want to mimic it in a small scale, but you want to prove that your scale down model is also representative of your commercial scale model. So this is something very important because this scale down model will be used as a tool for doing a lot of studies that you cannot do them because of the financial burden in a commercial scale arena, especially when you're working with a CMO. So these are studies that you can do in small scale instead of large scale. And please remember that no scale down is perfect. We have equipment limitations, we have technology limitations, but on the end of the day, your scale down model should follow the same manufacturing principles as your commercial scale model, and on the end of the day, you should get the same. Your release specification should meet all its ranges in a perfect manner. That is something that you cannot waver out. And the scale down model, as I said, will be used for many, many studies. One of them is the DOE that we will talk about in a second. So please remember, it takes time to procure the equipment. Today, bioprocess equipment has lead times of between 6 to 12 months. It's a huge amount of time when you're in a hurry. So please, you need to do the calculations. You need to understand the limitations of the equipment, and this takes time. So please take that into account. This conceptual slide will just explain a few connections between FMEA that we talked before and a scale-down model and the potential critical process parameters and DOE studies. So we'll make this very simple just for you to understand. And this is the basic idea behind quality by design. So we're going to give it 2 minutes, not any more than that. So we said that we are using the FMEA, the failure mode effect analysis, the risk assessment of the process, and high risk parameters will be considered as PCPPs, which is potential critical process parameters. So potential critical process parameters are potential until they are not proven to be fully critical. And these are also parameters that could affect the safety or efficacy or quality of the final product. So once we chose our 6 or 7 potential critical process parameters, and once we have established our scale-down model, what we can do is combine these two issues, the scale-down model and study the potential critical process parameters in a DOE design. So a DOE design is, in simple words, is an experimentation where we take the potential critical process parameters and challenge the system. So if we just take a look at the table on the right lower side, we see here, for example, culture temperature, where we work in 37 degrees. We could challenge it, the process, what happens if we work in a low range of 35 degrees or in a high range of 38 degrees. The same thing, if we decided that the DO was a potential CPP, we could decide to challenge the process with the lower range of 20% DO or the higher percent of 50% dissolved oxygen. And the great power of DOE is that when we go to a person that is an expert in DOE, he could do a DOE design, which by his special design, he can give us in 7 or 8 experiments knowledge that is equivalent to maybe 72 experiments. And not only that, we could learn in these experiments if there are interactions between parameters, if there is an effect on the temperature on the DO, if there is an interaction between the effect of pH on agitator speed, and so and so. That is why DOE is considered a very valuable tool, because in a very narrow amount of experiments, you get huge amount of data and understanding of your process. And last thing I would want to say, this QBD design does result in a QBD space, and this could help you in the future with deviations, meaning as long as you're in a validated design space, you can easily solve the deviation, and it will help you in the future release batches. So this QBD has also implications on your commercial scale, on the stage that you're in commercial scale, and could help you even in deviations. So, kind of approaching the last maybe 20 or 30% of the presentation, I would want to talk with you guys about engineering runs. So what are engineering runs? Engineering runs are the commercial scale runs that you actually perform in your commercial scale before doing the process validation. And these at-scale runs are very, very important. Why are they so important? Correct? They are costful. They are a burden from a financial point of view, but they are the most critical stage for accumulating real process knowledge at your real and final scale. So this is a great opportunity for you to know how your process performs in the final scale that will be used for process validation and later for commercialization. It is a key opportunity for process improvement. You did an engineering run and you saw issues that could improve your process. You saw issues where you had failures and you must improve your process. This is the time to do that, because when you reach process validation, your process is locked. You do it 3 times or more according to what you decide, but it's too late then. So please give yourself the chance of accumulating your knowledge by doing 2, 3, 4, or 5 commercial scale runs. Remember, these commercial scale engineering runs could also be used for making your material for phase 3, even for phase 2. So on the way, on the same time that you are preparing the material for your phase 3 studies, you're doing these engineering runs, and you're accumulating this critical and important data. And as I said, this is the most important risk mitigation before going into process validation. This is what will provide you the confidence that you are on the right track. There is no regulatory obligation to do engineering runs. This is an internal risk that the company needs to run internally. And on a theoretical point of view, you could immediately go to process validation without doing any engineering run. But I don't know any company that did that till today, because process validation is a complex effort. It has many, many sides, and you want to make sure as much as possible that you're ready. So I'm not saying that you will do 15 engineering runs, but try not to do one. So find your magic number somewhere in the middle, maybe towards the lower range, and to arrive to the place that you feel comfortable with your process. You can use your engineering runs to create stability data. So you could also have stability data. You finished your engineering runs, you made your drug substance, you made your drug product. You can start accumulating the stability data. And if you did not change the process, and meaning the process that you used for the engineering run and the process validation run was the same, this stability data could even be used for submission and to provide you already supportive data, and later you could complete the rest of the required 3 process validation batches. And in addition, the engineering runs is a great opportunity to do your cleaning validation. Now, this is a sensitive issue because a lot of companies, when they do not have time and they are pretty rushing with their clinical development, they find themselves doing the cleaning validation in parallel to the process validation. It's okay, but I would say this is not recommended. The recommended or preferred situation is that you complete cleaning validation before you go into process validation. And this can be done if you will do your cleaning validation on the engineering runs, have it closed, and then you're fixed. You go into process validation, and you do not need to worry about cleaning validation. Please take that into account, and please take care that if you're going into process validation and one of the batches that you did process validation failed cleaning validation, you will have to carefully decide if this batch could be released. So you could succeed in process validation. You have a check mark on that, But if you failed in cleaning validation, it might raise the question if the batches that you actually manufactured during process validation could actually be released. So please take that into account. For more information on that, you could also contact Adres. We will be very happy to help you. The last slide, which might be the most important slide, is something that all of us or most of us hate, which is Gantt. Gantt, 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 Gantt. Okay, so Gantts are a nuisance. We don't like to mess with them. Why? Because they take a lot of time. It takes a lot of time to set them up. That's right, not to manage them. Sometimes they put reality in our faces, and maybe we prefer not to see that reality. But on the end of the day, it's a big, big, big mistake. Why is it a big mistake? And this is, you have to understand that Gantts have a great power by providing you a retrospective look on why your commitments to finish something in half a year took 1 year. You could understand why you had creeping delays. You could identify critical paths. And this is very, very important. You want to have your resources obligated to the activities that will delay you, not on the activities that will not delay you. So you can prioritize your resources on that. And the other issue is that you could understand the interactions and dependencies between activities. If activities are dependent on each other or are not dependent on each other. So if the VP operations or the VP R&D or the CEO comes to you and tells you, okay, I understand that this study will be delayed by 1.5 months. Could you please tell me how will that delay the end of our project in a year from today? Without a Gantt, you will never be able to tell him that. So try to use a resource in your company. I don't think that for a startup, you need a full-time employee, but a person that could manage a Gantt platform. It takes a lot of work, so maybe it will take him a few days to set up all the activities, to interlink them correctly, but it will be worth the effort because on the end of the day, once a month, he will meet with the team, he will update the Gantt, and you will understand where you are, why you are delayed, what is now your critical path. And please remember, critical paths change, and they can change on a at least quarterly basis. You think that your DOE was the issue that delayed you from going into process validation, and after 3 months, you find that it's your engineering run. So this is very important to understand. In addition, it will provide, as I said, on the end of the day, increased control and understanding for the senior management, and providing your board or your management a commitment on timelines without having a Gantt will be completely irresponsible. Completely irresponsible. Because then you're just throwing a number. And anyway, these are always estimations. So a Gantt is not something sacred. You cannot bend times in space according to the Gantt, but it will be the best possible estimation that you are able to give at that certain time point. So please remember, no one is a prophet. No one can promise anything. But if you're promising timelines or promising commitments on finishing activities without a Gantt, you are totally providing promises that are not connected to anything. So this is in general what we wanted to talk about today. You are welcome to provide questions in the chat. If you, and as Tanya said, if you forgot to ask a question now and you want to ask this question later, you are more than welcome to approach us to the mail that Tanya is showing here on the presentation. We will be more than glad to help you, to support you, and use our expertise to help you move in the right direction. And the most important things is doing the right things on the right time, because we know startups, we are short on resources, we are short on time, and we are short on budget. So let's make sure that we do the right things at the right time. So that's my main takeaway message for the people attending today in this discussion.
We have no questions,
so if you do not have any questions, first, if you enjoyed this webinar today, we would like you to tell us if you enjoyed it, and please take your time to think about the things that have been discussed today, and you can contact us, and we will make sure to contact each one of you personally for each question that you have. So thank you for joining us today in this webinar, and Adres will in the future have additional webinars that will help you in various facets of clinical development, focusing you, providing you directions in places where you have questions and uncertainties. So thank you very much.